All right. Well, I'm really excited to introduce Dr. Kelly Miller. So Kelly obtained her bachelor's degree from Indiana University in Bloomington in psychology and animal behavior. She then went on to obtain her master's and PhD degrees at the University of Memphis in Tennessee.
Thanks.
And I'm really happy to announce that she has joined the Department of Biology at Ball State University, and she started her tenure with our institution in the fall of 2025. And she's currently an assistant professor in our department. So Kelly is a field ornithologist and a behavioral ethologist, and she uses nest boxes to learn about the breeding biology of wild birds. And so tonight, what she'll try and do is unravel some of the mysteries in terms of how do female birds make choices in terms of which male to mate with. So with that brief introduction, I'd like to ask Kelly to come up, and her presentation title is Good Genes or Good Parenting, What's Preferred in a Bird's Mate. Okay, please welcome Kelly.
All right, thank you, everybody. Let me make sure that we are situated here. Okay. Just not going to touch anything and hope that this continues for us. I am very excited to be here presenting to you all tonight for my first time with a group of fellow bird lovers, but also particularly excited to present about this project. This is actually one of the first experiments that I conducted as part of my graduate work at the University of Memphis. And it's one of those projects that has been kind of a doozy to get to the final step of publication. It took longer than it probably should have during my grad program to actually get the manuscript written up. But then the past couple of years, we have been kind of in a battle with a particular academic journal that shall not be named, going through 2 rounds of reviews and revisions to ultimately get rejected. But it's in the past now. The good news is we recently submitted to the journal Animal Behavior, and as of a couple weeks ago, this work was accepted for publication with minor revisions. So hopefully it will be out very soon. Thank you. So that being said, my talk tonight does really focus on the research, but I'm more than happy to talk about kind of my undergraduate studies, how I got to be in this position, if anyone is interested in that. Yeah, I forgot about our bird clock in here. Oh, okay. So really this research starts with this kind of dichotomy between males and females, where females tend to be the choosy sex, being more particular about the type of male that they mate with, and males are the competitive sex, competing against or amongst one another for access to mates. And really this choosiness comes down to a couple different things. First, it's related to just the costs of reproductive investment. So if you consider the initial investment by males and females in reproduction in gametes, just egg cells and sperm cells, egg cells that females produce are much larger. They contain all of the organelles necessary for cellular function. And for birds, they also contain the yolk, which provides nutrients to the offspring as they're developing. On the other hand, sperm are much cheaper to produce. For many bird species, with the same amount of energy and resources that it takes a female to produce 1 egg, males can produce hundreds, if not thousands, of sperm cells. So that initial reproductive investment is much greater for females, which leads to them often being the choosier sex, being more particular about the type of male that they want to kind of devote this investment to. This choosiness is also related to the factors that limit the fitness of each sex. So really this comes down to Bateman's principle, which states that female fitness is limited by the number of eggs that a female can produce, while male fitness is limited by the number of mates that a male can obtain. And that's what's shown in the graph here. On the x-axis here, we've just got the number of mates that an individual obtains. On the y-axis is relative fertility, or reproductive success is another way you can think of that. So for females in our dotted line here, you can see that they do. Is this the clock or is this? Okay, okay. I'm wondering why it's still going on. All right. So for females here in the dotted line, their reproductive success or fertility, of course, increases once they obtain one mate. But for females, obtaining additional mates doesn't necessarily correlate to an increase in their reproductive success. For males, on the other hand, this relationship is essentially linear, where the more mates they obtain, the more their reproductive success can increase. So really, this greater initial reproductive investment by females, plus the ability to obtain mates being the limiting factor of fitness for males, leads to this dichotomy where females tend to be choosier, being pickier about the type of male they mate with, and males being more competitive, competing against one another for access to potential mates. And it's thought that female choosiness is really the driving force of sexual selection on a male's phenotype, because these traits that are preferred by females can proliferate throughout a population if those traits are heritable, and females are more likely to mate with males that have the trait compared to those that don't. So in our scenario here with our male that's been chosen by this female, let's say, for example, that this male differs from others in the population because he's got longer wing length, which will be the main thing I talk about with this project today. So over time, over generations, we might start to see if longer wing length is a heritable trait, that this population starts exhibiting these males with longer and longer wings if females really prefer that trait in their male. But what do these preferred traits actually indicate? So often these traits are condition dependent. So they're an honest signal of the male's quality or condition. So here there are a few different hypotheses to describe this relationship. But for my talk tonight, I'll focus on the good genes hypothesis on the one hand, which essentially states that these traits that are preferred by females tell her that this male has really good genes that he'll pass down to his offspring, and maybe those genes confer greater survival or reproductive success for those offspring. So in this scenario, offspring inheriting a male's good genes is an indirect benefit to that female because she doesn't benefit immediately. Those offspring have to inherit those good genes and then go on to produce offspring of their own before the female really sees any fitness benefit. On the other hand, the traits that are preferred by females might indicate that those males will be a really good parent. And this is also called the good parent hypothesis, essentially stating that these traits in males are positively correlated with their parental abilities. So a male who has a better trait might also invest more in her offspring. And this is a direct benefit to the female because if those offspring are receiving more care from the male parent, they'll be more likely to survive. And she might not have to invest quite as much in those offspring. So she'll also be able to retain some energy and resources to maintain her own body condition and maybe go on to produce more broods in the future. So mate choice can influence parental investment decisions. And here there are two major hypotheses to describe this relationship between mate quality and parental investment, one of which is the differential allocation hypothesis. So for this hypothesis, if we have a mated pair where one individual is lower quality and the other individual is higher quality, we're expected to see the lower quality individual significantly increase the amount that they invest in those offspring, essentially because they recognize they're paired with this really high quality mate. So they want to try to make the most of that mating attempt. At the same time, this high quality individual will significantly decrease the amount that they're investing in those offspring because maybe they recognize that they've mated with kind of a dud who's not as high quality, and they can increase their reproductive success by spending time doing other things like going off to find additional mates, for example. So another hypothesis related to mate quality and parental investment is the reproductive compensation hypothesis. I'll probably just refer to this as compensation, where again, if we've got a low quality individual and a higher quality individual, this time it's the higher quality individual that significantly increases their amount of investment in those offspring, essentially to try to compensate for any suboptimal genes that might have been passed on to the offspring by the lower quality mate. And in this scenario, the lower quality mate doesn't necessarily change their amount of investment in offspring beyond what would be expected. So this can also present a conflict between parents about how much either one should invest in those offspring. And I've been talking a lot about parental investment already. So just to give you guys more of a definition here, really it includes any type of investment by a parent in its offspring that increases the chances that those offspring survive, but at a cost to the parent's ability to invest in future offspring. So those costs and benefits with increasing levels of parental care are plotted here. The benefits in the blue line, you can see that they increase really rapidly with increasing levels of parental care, at least initially, but they eventually reach this plateau where it's diminishing returns on the parent's investment. And by investing more in those offspring, they're not really getting a gain in benefits. But the cost to future reproduction in this red line increase almost linearly with increasing levels of parental care. So really where parents want to be is somewhere around this dotted vertical line in the middle of the graph where the benefits exceed the cost by the greatest extent. And when we're talking about parental investment, really it's going to depend on the species we're talking about and whether we're considering pre or post-fertilization investment. But for birds and for post-fertilization investment, which is the focus of my research, it can include a number of different things from the number of eggs a female produces, the amount of time that a bird spends sitting on those eggs or sitting on their young to keep them warm. The really big one is nestling provisioning. So how much food are parents bringing to feed nestlings at the nest? But it can also include things like nest sanitation, nest defense from predators, competitors, etc. But again, parental investment really comes down to these trade-offs between investment in current offspring versus potential future offspring. And I always like to think about these trade-offs in the form of a pie graph because individuals are working with a limited amount of resources to begin with. So the more that a bird might invest in their current offspring, the less they might have left over to invest in future offspring and their own survival. So really, birds should strategically invest based on the conditions that they're experiencing at the time. So these conditions that might alter an individual's level of investment could be environmental conditions, abiotic factors like temperature, precipitation. They can also include ecological factors like food availability, or they can be social conditions, things like population density, the number of competitors in an area, or for the basis of this study, the quality of one's mate as well. So of course, high quality mates are going to be preferred, but they're not always available or attainable in nature. So the ability of an individual to alter their level of parental investment in relation to the quality of mate that they were able to obtain is adaptive and might help them kind of optimize their investment in current versus future reproduction. So we collected some preliminary data, actually before I even started graduate school, on a population of Prothonotary warblers, where we found a potential female preference for longer wing length. So in this initial year of data collection, we found that males with longer wings obtained more unique female mates over the course of the breeding season. We found that these males were more likely to pair polygynously when they had longer wings. And this just means that these males had active nests with two different females at the same time. We also found that the young produced by these males with longer wings were in better condition before fledging. So this could mean that those offspring inherited good genes from those males with long wings. Oh, we've got a deer friend. Sorry, it is kind of distracting, but really cool. So these nestlings being in better condition, could indicate that they inherited good genes from these males with longer wings, could maybe indicate that those males with longer wings are better parents and investing more in those offspring, or it could indicate that differential allocations occurring and these females paired to males with long wings recognize that they have a good mate and are investing more in those offspring, and the offspring are benefiting. Now, interestingly, with a trait like wing length that's directly related to a bird's ability to get around, get away from predators, related to their survival, we wouldn't necessarily expect it to be under strong sexual selection, but rather under stronger natural selection because it is related to the survival of these birds. But traits like wing length can also be under sexual selection if they're an honest signal of that male's quality that females can reliably use to help increase their reproductive success as well as the reproductive success of those males. And for Prothonotary warblers in particular, as far as their courtship goes, we know that males kind of spread their wings and flutter them as they're displaying to females. Everything I've read about this courtship display says that the females are normally perched above the male, kind of looking down on him while he's doing this wing display. But I have scoured the internet trying to find a video of this courtship dance. And what I have up here is the best I could find, and it's not terribly zoomed in. So I'm sorry in advance for the quality. But hopefully you'll be able to see that this male right here is kind of walking around on top of this rock, fluttering his wings. Interestingly, female's not above him, but you'll see her kind of follow behind him and assess him from that position. There's the male displaying. You can sort of see the wings spreading and fluttering here. But she's looking at him very closely to see what's going on. But yeah, and that is that. So after showing that, if anyone in here is a bird photographer or videographer and happens to get a video of Prothonotary warbler courtship, please share it with me. I would be forever indebted to you. Another interesting thing that we've observed in this population of warblers is that despite males and females having very similar skeletal sizes, males have disproportionately longer wings than females, so about 6% longer than the female average. So together, this suggests that sexual selection might be acting on this trait, but really the goal of this study was to get to the bottom of what's going on when it comes to mate preferences and how wing length might be altering parental investment. So I wanted to, after collecting those preliminary data, to conduct a manipulative experiment. And for this experiment, I hypothesized that male wing length would shape mating success and parental investment, and that by manipulating a male's wing length, we would cause behavioral differences in both the male and female of a mated pair, potentially following one of these hypotheses I spoke about earlier. So differential allocation, where the lower quality individual invests more, higher quality individual invests less in those offspring. Compensation, where the higher quality individual invests more to compensate for pairing with a lower quality mate. Or the good parent hypothesis, where essentially male wing length, if it's related to his parental quality, we might see a positive relationship where males with longer wings invest more in their offspring. So I've already mentioned that my study species was the Prothonotary warbler, but to give you some more background information about this species, they are a neotropical migrant. So you can see they breed throughout most of the eastern US, conveniently with a bald spot in their breeding range in central Indiana, that it is my life's mission to prove this map wrong. Really hoping to get some of those at our field site here in Muncie. But they do overwinter down in kind of Central America and northern South America. And just a fun tidbit of information for some fellow bird lovers here. I had some colleagues who were able to put geolocators on Prothonotary warblers on their breeding grounds all over the eastern US. And regardless of where these birds bred, about 90% of the population overwinters in northern Colombia, which is pretty neat to know about. Yeah. So in this species, males and females are considered to be sexually monomorphic or look very similar, but there is some difference in the brightness of the crown or the top of the bird's head, where males are much more bright yellow compared to the kind of olive green color of females. At the site where this work was conducted, females typically produce 2 clutches per season, so they do have the ability to mate with potentially more than one unique male, and they will produce more clutches than this if one of their clutches has failed. They're considered to be socially monogamous, so it's one male and one female that have a pair bond and care for a brood of nestlings. But we do see polygyny every once in a while where males will have active nests with 2 different females at the same time. Also, extra pair mating can happen in this species where the females go off and sneak a copulation with a different male. And so the male that's actually caring for her offspring might not be their genetic father. But they do exhibit biparental care of their young, where the female is the only one that contributes to incubation and brooding, but both the male and female will feed nestlings after they hatch. So this work was conducted again as part of my grad degree at the University of Memphis. We've got a field station down there called the Meeman Biological Station. It's located just outside of Memphis, but still in Southwest Tennessee, primarily secondary deciduous forest, as you can kind of see in these pictures. But we do have some mixed forest habitat where there are a lot of pine trees in certain areas of the site. But over this site, we've got 625 nest boxes spread out over about 170 hectares. And the nest boxes look like what you can see here. And lucky for us, they are readily accepted for nesting by the Prothonotary Warbler. So during the breeding season, we check all of these nest boxes regularly, so we're able to monitor what's going on in any given box at any given time. But we check them every 5 days, so we're able to determine the clutch initiation date for each nest. So for a small bird like the Prothonotary Warbler, females are laying 1 egg per day until they reach their clutch size of usually 5 eggs on average. So by checking boxes every 5 days, we can catch new nests and determine when they laid the first egg in that nest. We also collect data on clutch size, or just the total number of eggs that females produce. Once we expect these eggs to hatch, we'll start checking the nest more frequently so we can figure out the day that the nestlings hatch, so we know exactly how old they are. And then we track them throughout the nestling cycle as well. So we'll know the proportion of young that successfully hatched based on that original number of eggs in the nest, the proportion that fledged or successfully left the nest, and the date that they do that. Of course, we collect a lot of information from the birds as well. So all birds that nest in these boxes, all adults at least, are captured usually midway through the incubation period. Female Prothonotary warblers are the ideal study specimen. There were so many instances where I actually walked up on a nest box and made eye contact with the female Prothonotary as she was sitting in the box incubating, and I was still able to capture her. So super easy to study, at least for the females. The males, since they don't incubate, they don't spend a lot of time in the nest box, they're a bit harder to capture. So we have to use mist nets. We just usually set our nets up directly in front of the nest box. We'll turn on an MP3 player with some Prothonotary Warbler song to kind of lure him in. And they are fairly territorial, so usually that works pretty well. But we started to observe over the last few years that I think the males were getting used to the specific MP3 that we used. And so it became harder and harder to capture them in our nets. So a couple of years ago, we started using this decoy, which is just a wooden bird painted to look like a Prothonotary Warbler. We affectionately named him Lil Buddy. And we would put him out, and he worked phenomenally well. So you just set him on the top of the nest box with your mist net up. And there were many instances where we would turn around to walk away from the net, and before we could get 10 feet away, the male was already in the net. There were times when the males would fly in and actually knock Lil Buddy off of the box into the net with them. So that ended up working really well for us to be able to collect morphological data from these males. But all of the birds, the adults as well as the nestlings that are produced in these boxes, are banded with a uniquely numbered leg band, which you might be familiar with if Kamal has ever talked about his research here. But essentially, these leg bands give the birds a social security number, so we know exactly who each individual is. And if we ever catch an adult later on, we'll know exactly where they've nested before, who they've nested with, when, what their reproductive success looked like in these previous nests. And for nestlings, we'll know exactly where they hatched from as well. Adults will receive 3 colored leg bands, which we arrange in unique combinations so we can later visually identify the adults without actually having to physically recapture them each time to figure out who's nesting where. And we collect a lot of morphological data from these birds as well. So we'll collect tarsus length, which is just the length of the lower leg bone, which we collect using dial calipers as shown here. That's the bird's tarsus. We'll collect their wing length using a flattened or a stopped ruler, sorry. And wing length, that's how I'll refer to it in this study, but technically it's flattened wing chord, the way that we measure this. And we also collect data on the bird's mass using a digital scale, as this freshly hatched warbler is demonstrating here. And in many cases, to kind of better assess a bird's condition and control for their skeletal size when we're considering their mass, we'll do a linear regression of their mass and tarsus length. So birds that have a greater mass than we would expect given their tarsus length would fall above this line. Birds in worse condition with the lower mass than expected given their skeletal size would fall below that line. So if I ever refer to condition throughout this talk, that's really what I'm referring to. And although I do consider myself a field biologist, there is quite a bit of lab work involved with this research as well. So when we capture birds or band nestlings, we'll collect a small blood sample from their brachial vein in their wing, and we'll bring that blood sample back to the lab. We're able to extract DNA from it. So you might be able to see right here a clump of bird DNA in this tube that's been extracted from a blood sample. And ultimately, we'll use that DNA to do some lab work to determine nestling sex, because you can't really tell if a nestling is male or female morphologically while they're that young. And we do know that male nestlings tend to grow a little bit faster in this species and reach a larger size compared to female offspring. So we want to be able to control for that. And then for this study in particular, we also performed some paternity analyses thanks to our collaborator at the Field Museum, Dr. Dylan Maddox. So we're able to determine how many offspring produced were from extra pair copulations. So getting into the experimental manipulation, this took place over a 2-year period from 2018 to 2019, where we manipulated male wing length by clipping off the very tips of his primary wing feathers. So if we zoom in, trying to use the clicker, zoom in on this picture here, we can see male's wing length a bit closer. So with this manipulation, we created 3 different treatment groups. We had control males that were completely unmanipulated. So this male's wing length is falling right about 72 millimeters. So he would stay there if he was in the control treatment. Then for our experimental treatments, we had what I'll call the clip 2 males, whose wings were shortened by 2 millimeters. So this guy with a wing length of 72 would now be down to 70. And we had another experimental group that I'll call the clip 4 group, where their wings were shortened by 4 millimeters. So that would bring this guy down to 68 millimeters. And we chose these amounts to clip from the bird's wings because it corresponds to 1 and 2 standard deviations from the population average wing length. But we wanted to clip off the smallest amount possible, so it would still be an ecologically relevant amount of variation in male's wing length. And importantly, we performed this manipulation after pairs had already formed and produced a nest. And this really allowed us to kind of decouple the male's attractiveness from his quality and really test these hypotheses that we're interested in that relate mate quality to parental investment. So another major part of this study was to record parental provisioning effort, how often parents are feeding their offspring. So we did that by recording outside of our nests. So essentially we would go to the nest the day before we filmed, place this pole in the ground that has a cell phone holster duct taped to the top, really high-tech stuff here. Place this in the ground about 1 meter away from the box just so the birds could habituate to its presence. And then the day of filming, we only visited the nest to count the number of nestlings in the box and then start recording. So with these videos, I assessed an hour of parental care, and these videos were recorded when the nestlings were 4 days old. So if we look at this graph that shows nestling body mass in relation to their age, 4 days old falls right during the period of fastest nestling growth. So presumably parental care during this time is really important for future nestling success, since that's when they're growing the fastest. They need the more resources to be able to grow to their ultimate size. And with these videos, I just quantified the number of trips that parents made to the nest with food to feed their nestlings. And I was able to do this for 109 nests over that 2-year period. So this is an example of what these videos looked like. It'll happen really quickly. When I was scoring these, I had the opportunity to slow it down frame by frame so I could really see what that food item was that parents were bringing, if they were bringing food at all. But what we'll see in this video is a parent Prothonotary comes and lands in the nest box entrance here. You'll see it kind of move around a bit, and it'll very quickly turn to look out the direction the front of the box is facing twice. And if you look really closely, you'll be able to see a big green caterpillar in its bill. So there's the bird. Right there. Again, happens really fast, but brings that food to feed its nestlings, and it will record the time that the parent arrives at the nest, whether or not they have food when they leave. So we also know how much time they're spending in that box as well. So this study really spanned an 8-year period. So I mentioned the preliminary observations that we collected in 2017 that kind of gave us the idea and informed our subsequent manipulation, which took place in 2018 and 2019. And then we continued monitoring these birds through the 2024 breeding season, collecting data on male morphology, mating, and reproductive success. So really, again, an 8-year data set where we had 836 nests produced by 208 unique males at this site. And we were using this larger data set, kind of going back after our manipulation to revisit some of our initial questions, and particularly to investigate the role of male age in both his wing length as well as his mating and reproductive success, because that was something that we were limited by with our preliminary observations. I will also admit that we didn't really know how to age these males for most of the time during this study. We figured that out towards the later end of the study, but in general, when we had a new male arrive at our site that we hadn't banded before, we just assumed he was 1 year old. And I will say that now that we've figured out how to age them more accurately, that is actually true in more cases than not. But I would have liked some more detailed data on male age. So we were able to look at that a bit more in depth with this 8-year data set because we had more males that were returning to the site over multiple years, as well as some offspring that hatched in our boxes and then recruited into the breeding population. So very briefly, just to touch on some stats that were performed in case there are any statistics people in here. We standardized all data prior to analysis just to ensure consistency and kind of facilitate our interpretation of our results. But we primarily use just linear and generalized linear mixed models. I did include nest ID and parent ID as a random effect in these models whenever we were looking at multiple nestlings within the same nest or multiple broods produced by the same parents, just to control for any individual variation there. And I also included covariates in all of the models that I'm presenting results from today to control for these things that we expect to contribute to variation. So when we were looking at wing length, we're also always controlling for other aspects of the bird's morphology, like their mass and tarsus length. When we're looking at provisioning, fledging success, nestling condition, we're controlling for time of year because we know that over the breeding season, food availability typically declines. So that can impact how often parents can bring food to the nest and also the condition of those offspring. We controlled for brood size in those analyses as well, just because we know that larger broods means parents usually bring more food because they need to take care of more nestlings. But also that can negatively impact nestling condition and potentially fledging success as well. And finally, we did some analyses of heritability where I also controlled for nestling sex, since I mentioned that we know that male nestlings might grow a little bit faster than female nestlings. So first, to get into the effects of the manipulation itself. So just a quick reminder, we had our control males that were unmanipulated. They had the longest wings. We had our clip 2 males that had their wings shortened by 2 millimeters, and clip 4 males had their wings shortened by 4 millimeters. And here we're just looking at male wing length, both pre and post manipulation, and no difference between our males pre-manipulation. Post-manipulation, we see this expected difference where all of the groups now significantly differ in wing length, with our control males, again, having longest wings, clip 4 males having the shortest wings. Now, because we're manipulating wing length and we know that this might impact a male's ability to get around, we used an established formula that looked at a male's mass versus his wing length to calculate flight costs, just the amount of energy that males have to expend in flying around. And here we did find an effect of our treatment, particularly for our clip 4 males that had the shortest wings, where after manipulation, they now also are experiencing significantly greater costs in flying. We didn't necessarily expect to induce this significant difference, but because we found this, we did want to verify that we weren't negatively impacting male survival. So we looked to make sure that our treatment didn't negatively impact a male's ability to breed again within the year he was manipulated and in the next year, and we found no effective treatment there. So we might have slightly increased the amount of energy males required to fly around, but overall not his survival. And I will also add that these males molt their wing feathers before they migrate. So we shouldn't have negatively impacted their migration ability either. So getting into some mating effects of this manipulation, we didn't find a whole lot here when we looked at mating, but one interesting thing we did find is that our control males that had the longest wings were more likely to be divorced between breeding attempts compared to our experimental males. Now, I will say we kind of have to take this finding with a grain of salt because we only had 4 instances of divorce between breeding attempts, but all of those occurred with our control males. Our experimental males were never divorced. Females tended to keep them as mates for a longer period of time. Otherwise, really no effect of our treatment on mating. So no difference between our groups in the number of mates males were able to obtain, in the probability of them pairing polygynously with multiple females at once, or in the likelihood of females producing extra pair young when paired with these males. Now, although we didn't find an effect on extra pair copulations in relation to our treatment, we did want to see how males' natural wing length might impact the likelihood of females sneaking off to find another male. And we found an effect here. So when males had longer wings naturally, they were more likely to sire extra pair young compared to males that had shorter wings. So this suggests that there is some benefit to females of pairing with these extra pair males, potentially a genetic benefit in obtaining their genetic information through these extra pair copulations, but maybe not a parental benefit, since we found that our control males in our experimental manipulation were more likely to be divorced and not mated with again. And we think we found that finding because we found an effect of our treatment on male provisioning. So essentially, this was part of a treatment by parent-sex interaction, and there's a lot going on here, so I'm going to walk us through it. The first significant effect we found was a difference between our control males and our CLIP4 males, and similarly, a difference between CLIP2 and CLIP4 males, where in both of these cases, those CLIP4 males that had the shortest wings were feeding their offspring significantly more often compared to our other males. We also found a difference within that CLIP4 group where those males were provisioning more often than the females that they were mated with as well. So this shift toward greater paternal investment with shorter wing length could be due to a couple different things. On the one hand, we might have kind of changed males' foraging behavior with our manipulation. So there was another study that purposely handicapped males and actually, or not just males, but all birds, and actually removed flight feathers to observe the effects on foraging behavior. And this study found that those birds weren't foraging any less after that handicap, but they were more likely to forage closer to their kind of home shelter or home base. So if that's the case in our males, maybe they were more likely to hang out closer to their nest rather than drifting farther away. And that might have made them more attentive to nestling begging calls, so maybe increasing their provisioning that way. Another explanation here could be that these males with shortened wings recognized a reduction in their reproductive value. So we may have triggered a form of kind of terminal investment in their offspring where they're unsure if they're going to have the chance to reproduce again in the future. So they want to make the most of their current breeding attempt. Third explanation here, it could be differential allocation by the males. So if these males recognize that they're now lower quality than the females that they're paired with, in differential allocation, it's the lower quality mate that increases its investment in those offspring, while the higher quality mate decreases their investment. So that could be what's going on here as well. But because we didn't find a significant difference within our females between these treatment groups, we don't think that females were recognizing a phenotypic difference in their mate after this clipping manipulation. We think that this female or these females paired to our CLIP4 males were just responding to an increase in their mates' provisioning levels. So since their mates were provisioning more, they decided to kind of try to hang on to some resources to maybe maintain their own body condition. They recognized that they didn't need to invest as much for those offspring to survive. The next thing that we found was an effect on fledging success in relation to our treatment, where in particular, our experimental males fledged a greater proportion of their offspring compared to our control males. And we really think that this difference was driven by that difference in male provisioning. So here we see male provisioning on the x-axis in relation to, again, proportion of young fledged. This is just based on the number of eggs in that nest. And we see a highly significant positive relationship here, where when males provision more, they're able to fledge more of their young. And this finding has been found in the literature before. Really, male investment is the limiting factor on nestling success because male investment is so much more variable than female investment is. So when males are investing more, their offspring are more likely to survive compared to when they're investing less. And similarly, we found a significant positive relationship when we looked at male provisioning in relation to the proportion of their young that actually recruited into the breeding population. So these are offspring that were hatched by these males in a previous year and then returned to breed as an adult in the population in a future year. And again, just re-illustrating this positive effect of male investment here. We also wanted to examine recruitment in relation to nestling traits to see if there's anything about these nestlings that might make it more likely for them to survive. And in these analyses, wing length came out as the strongest predictor of recruitment. So essentially, nestlings, when they attained longer wing length prior to even leaving the nest, they were significantly more likely to be seen in our breeding population in a future year. And if you think about these nestlings, 1 to 4 months after they leave the nest, they have to migrate to their overwintering grounds. So it's possible that if they're able to attain longer wings before they even leave the nest, maybe that makes this long flight more efficient and helps them survive that journey. So since we found this effect of nestling wing length on their probability of surviving, we also wanted to try to examine the heritability of this trait. So here we're looking at narrow sense heritability in the form of mid-parent wing length, so just an average of male and female wing length. And here we're only considering natural wing length, not as a result of our manipulation. And we did find this significant relationship here between parent wing length and nestling wing length prior to fledging, although somewhat weaker, but still significant. And I will point out for this graph that all of the data here were from nestlings' genetic fathers, but we ran the same analysis with social fathers instead and found qualitatively similar results. We actually only ended up having about 8% of young that were produced by an extra pair mating in our population over this 2-year period. So potentially some heritability that we see even during the nestling stage in this species, but we also looked at heritability for our offspring that recruited into the breeding population as adults. And here we find a bit of a stronger relationship, still significant between mid-parent wing length and the wing length of these offspring once they reach adulthood. I've got this graph kind of split out between sons in the black circles and daughters in the white triangles, just so you can see the differences in the sexes there. Positive effects on both sons and daughters in relation to their parents' wing length, but maybe a slightly stronger relationship for sons. So really these heritability results tell us that females might be gaining that indirect genetic benefit by mating with these males that have longer wings. If their offspring are more likely to attain longer wing length before fledging, which increases their likelihood of surviving to reproduce in the future, and particularly if they're producing sons that are inheriting the longer wing length of their fathers, which might increase their ability to obtain mates as well. So finally, we wanted to kind of revisit our initial questions from our preliminary year of data collection and really try to figure out what a male's wing length tells a female. So here we were able to look at factors like male age that we weren't able to look at previously. And we found a significant relationship here where as males get older, their wing length tends to increase. So for this figure, the point size here, these gray circles correspond to the number of data points that we have in that area. We've got a lot more data from 1-year-olds because these birds often only live a couple years and then we never see them again. But we do have some older individuals in our population, especially now that we've had these nest boxes for a longer period of time as well. So wing length tends to increase with age. So really when a male has longer wings, he might be telling a female that he's older and that he has a better genetic quality or a better ability to survive for a long time. We also revisited some of our initial analyses and found that male age was the strongest predictor of his initial breeding date. So this suggests that maybe those older males are more dominant. They are able to defend a territory or acquire a territory earlier in the breeding season compared to younger males. Age was also related to the number of mates that a male obtained and his likelihood of pairing polygynously. And because these older males were able to obtain more mates, they also produced more fledglings in any given year, so had higher reproductive success overall. So I know that was a lot of data to throw at you guys. So to just quickly recap our original hypotheses here, we did find support for the first hypothesis that male wing length shapes his mating success and parental investment. So looking only at our experimental data, we found that control males with longer wings were slightly more likely to be divorced between breeding attempts, but those experimental males with shortened wings that invested more in their offspring were more likely to be retained as mates, and those offspring were more likely to survive and recruit into the breeding population. With our observational data, we found that males with naturally longer wings produce nestlings with longer wings as well, and those nestlings were more likely to survive and recruit. Also, we found that these males with longer wings were older, and older males obtained more mates and produced more fledglings. For our second hypothesis, we found kind of mixed results here. So we did find that our manipulation of wing length created behavioral changes in males and females within the mated pair. But it's not totally clear. We probably need some more research when we're looking at these individual hypotheses. So as far as differential allocation goes, we didn't find a ton of support, but we did find maybe some evidence to suggest that our experimental males were increasing their provisioning effort after the manipulation. Their mates were decreasing their provisioning effort. So there might be some differential allocation going on there. But I think that needs a bit more research to say that with certainty. Really no support for the compensation hypothesis. We didn't really find a situation where the higher quality mate in a situation was investing more to kind of compensate for pairing with the lower quality individual. I would say the good parent hypothesis is the one that we found the best support for. So we did find that females were more likely to maintain pair bonds with experimental males who provisioned their offspring more, who were better parents. But this finding isn't totally consistent with the good parent hypothesis in its original form. So with this hypothesis, it's predicted that male traits and parental ability will be positively related to one another. So we should have found maybe that males with longer wings were better parents, but here we found that males with shorter wings were better parents. So I still think it's some support for this hypothesis, but not exactly, I would say. So really, we found that longer male wing length might signal greater genetic or phenotypic quality, and shorter male wing length might signal greater parental quality. And females might differ in the type of signal that they prefer to respond to in any given season, depending on the environmental conditions, her condition at the time of mating. All right, so with that, I've got a small army of people to thank at the University of Memphis, particularly my grad and postdoc advisor, Dr. Keith Bowers here, who allowed me to do this work in his lab, as well as all of our amazing fellow graduate students and undergrads and my sources of funding for this project. I also have to give a shout out to my current lab members at Ball State. A lot of them who were involved in the lab last semester actually helped with revisions on the manuscript that will be published here soon. But most importantly, they've been helping me establish a system of nest boxes at one of our properties here in Muncie, Genwoods, so we can start to answer some similar questions with local birds here at Ball State. And with that, I'm happy to take any questions.
Well, if there are any questions, Kelly, if you could just repeat them for those that are on Zoom with us. So
Yes.
Questions?
Yeah.
Yes, you said that males with greater wings, that their wings got longer as they age. But is that the case, or is it that survival of those with shorter wings?
Ah, that is interesting. A different way to look at it. So talking about the finding of males' wings getting longer as they age, the question is that the case, or is it that the males with shorter wings are just less likely to survive? I guess we don't know because we don't have those shorter wing males to potentially collect data on. But I will say, if I can find my mouse and get back, nope, hold on. If I can pull that graph back up really quickly. So with this data set, we did have a few individuals that we actually saw decrease in wing length over time as they got older. But the majority of the population increased in wing length. So that is really hard to tease apart. I might have to try to look into that farther.
Hey, other question.
EJ?
Will they change parental investment based on wing damage, like about
Oh.
damage, where like the flipping may have skewed it towards the differential because of he thinks that he's now damaged.
Yeah. For those on Zoom, question is, could natural damage to the wings potentially lead to changes in parental investment by the males? I think it absolutely could. It would probably depend on when that damage occurred and what type of damage. So if the bird fully loses its feather, it'll just regrow a new one. But if that feather is damaged, I don't know how quickly they would be able to molt that damaged feather out to replace it. So it could end up in this scenario like we saw with this study where it might change what the males are doing just because they maybe can't fly as well, changing their behavior and foraging, things like that. Absolutely. Yeah. Mhm.
When during the day you see the raised bird.
Oh, that's a great question. So this also comes back to, the question was when during the day do we see the most provisioning occurring? I want to say in the morning, but there have been studies kind of looking at provisioning over the course of a whole day, and they found that it's fairly consistent. So we only recorded provisioning for an hour, or we only scored an hour of the videos we recorded. All of those videos were recorded between about 6 a.m. and noon. So they should have been comparable in the sense that at least we're recording all these videos at the same time. But I actually have another data set that I'm sitting on and really need to try to get published, where we looked at this hour of recording to see if it's representative of what the birds are doing, even over shorter time periods, because we run into these issues with field research where maybe your camera dies or some weather comes in and you only get 20 minutes of recording. So is that still useful? And we've essentially broke our hour-long recordings down into 10-minute segments and extrapolated what we found in those 10-minute segments to an hour. And there was no significant difference between the shorter segments versus the full hour. And there are multiple studies out there showing that an hour should be representative of what's going on over the course of the whole day. So yeah, it seems like it's not changing too terribly over the course of a day. But still my tendency is to want to say that they're a bit more active in the morning compared to afternoon, and especially at night when they're resting more. Yeah.
Is there, did you randomly select individuals for deciding whether or not to do like living control? Because I remember you said that you wanted to make sure that it didn't like negatively impact their survival. So and like maintained in like the average range. So at that point, like, for the longer clipping before, would you choose like a male that had like longer wings already that would better?
No. So question is about how we applied the treatments, whether it was applied randomly, and it was applied randomly. So we basically flipped a coin to figure out what we were going to do for the first treatment of the season, and then we just went through and alternated between control, clip 2, clip 4. We didn't have equal amounts of birds in all of those groups though, because we ended up having some males that had already been treated that were mated polygynously, and so we were planning to do another treatment and then couldn't because they had already been treated, or a male that we captured thinking he was the male at a nest, but really he was just flying through. So he never actually produced a nest in our study. So that's why we ended up with not totally even numbers between the treatment groups, but in general, they were applied just totally randomly in order of what the last treatment was that we did. Yeah.
So where should we go? Closest area we can go to see these prothonotary nesting.
Yes.
And why are we nesting here?
Great questions. Question was, where do we go to see Prothonotaries close to here nesting? So I have been tracking religiously the Prothonotary sightings on eBird recently. Someone actually saw one at, I think, Fort Harrison State Park in Indy a couple days ago. I'm hoping that soon you'll be able to come to my field site and see them nesting here in Muncie. So keep your fingers crossed. I don't totally believe that range map and that bald spot. I think we probably have fewer Prothonotaries here than other areas because there is so much agriculture and they really like old growth forest. Something that's really interesting about this species is typically they're considered to be swamp birds living in like flooded forests. But my field site in Memphis was not that habitat at all. It was near the Mississippi River, kind of in the Mississippi River floodplain, but it didn't often flood. And we have some larger ponds at this site, some kind of small ephemeral streams. But these Prothonotaries were nesting in boxes just in the middle of the woods, nowhere near a body of water. So I like to think they're more of an if you build it, they will come species. But we'll have to see how it goes. Yeah.
That Prothonotaries will strip grapevine to put in their nests.
Ooh.
Is this true? Are you finding grapevine fibers in the nests of Prothonotaries?
Question is, are there grapevine fibers in the nest of Prothonotaries? Probably. I am not enough of a plant expert to know whether it's grapevine fibers versus some other type of tree bark, but I have seen material like that in their nest before. They like a lot of moss. They use primarily moss in their nest, but they will use grasses, bark, not usually leaves and not usually twigs per se, but they will use materials that are kind of softer like that. Nate. Yeah, it seems like that might be something we're doing this summer, and hopefully we'll get Prothonotaries to be able to assess.
That was the video data from flip 4 males. Can analyze yet to see if the prey type or size is, because you mentioned that there could be different strategies that they employ, you know, closer to the nest, possibly for foraging for prey, but could that also be dictating a difference in their prey choice? And does that nutritionally impact the
yeah,
so.
That's a great question. Question was about other differences potentially in provisioning of these males in the clipping experiment, whether they might differ in prey size or type that they're bringing to the nest. That is not something that we looked at. I mean, we've still got the videos. So if there is someone here that might want to look at that, you absolutely can. I will say with these external cameras with this setup, it's a lot harder to accurately determine. I think we could accurately, actually, I want to say when I scored these, I was determining prey size. So we probably could easily look at that. Prey type might be a little more difficult, but that would be super interesting. Yeah. Would be really interesting to look at that. Yeah, Kaylee.
Prey type.
They're eating.
Oh.
Feeding the young.
Ooh, that's a good question. About whether habitat differences might change preference for wing length. I don't know if it's universal. We haven't done this study in other habitat types to really be able to compare, but I will say that it probably just generally depends on what's available to the birds in the habitat. If there's a lot of food available, it's a really good year, the females might prefer those males with better genes over those males that will be good parents. If they're able to care for the offspring on their own, maybe they don't need that additional help from the male. But yeah, we haven't done a comparative study between different habitat types to be able to give you a really good answer to that question. Yeah.
So tell us a little bit about site fidelity.
Ooh. Yes, questions about site fidelity in Prothonotary warblers. They have very high site fidelity. So for the adults, we'll see often that they are returning to breed in the same exact box year after year. For the nestlings that recruit into the breeding population, they still have relatively high site fidelity. They, of course, don't return to the same exact box that they hatched from because they want to try to spread out a little bit, avoid inbreeding. But we do, at least at this site, our boxes are spread over a wide enough area that we do see a lot of those returning offspring. And I know there was one study that looked at how far these offspring were moving from the box that they hatched out of versus the box that they ultimately bred in. And I want to say it was within a mile from where they hatched in most cases. So still returning to a fairly similar area.
Okay, so a follow-up on that would be, did you have males that were manipulated since a 2-year study? Manipulated in the first year, did they show up again on the second year? And then did you use those same males to manipulate or not? And then the whole
Yes.
question is about pseudoreproduction.
Yes, great question. About whether we manipulated males again if they returned to breed in that second year of the study. We did not. We left them alone. We ended up analyzing the data to see if there was an effect of the treatment the previous year on male mating success, reproductive success that second year, and we didn't find anything. So I want to say we ended up for some of these analyses, including those males that returned that we didn't manipulate in the second year as control males, just because there was no effect of that first year treatment on them the next year.
All right, any other questions? I'm going to, So are there, well, I guess I'll, yeah. Are there any questions from those that are on Zoom?